Start the record. Okay, so we're now recording. Thank you very much for giving a Build a Cell seminar, and it's all yours.
All right. Hi, everyone. Thank you. First of all, I want to thank you all for coming before I even introduce myself. It's really nice to be able to speak to this group. I've admired it from afar for a long time. Let me actually go ahead and put my slides up so that we can all see them. I should have done more. Oh. All right. So can you all see these?
Yep.
Excellent. Okay. So yeah, my name's Shankar Mukherji. Again, thanks for coming. I'm an assistant professor of physics and cell biology at WashU. A little bit about myself. It was with Kate about as we were waiting for people to show up. I'm at home, so we have a whole gaggle of children downstairs. If any of them happen to come by, just say hi and sort of wave them off. My lab works on basically the problem of how cells allocate resources in order to grow. That's kind of our 30,000 foot view of what we're interested in. And the particular scale at which we're looking at this now, and what I'm going to share with you today, is at the scale of these mesoscopic structures in the cell, these organelles. So these are really large for the cell structures. And the reason that we got interested in this problem is maybe reflect my own background, which I got my PhD in Alexander van Oudenarden's lab. I studied gene regulation by microRNAs there, but of course, much of the lab studied the phenomenon of stochastic gene expression, which basically how it is that the process of gene expression is impinged upon by random forces and causes variability in gene expression. So I was very intrigued by that idea, as many of us are, I think. And in particular, what intrigued me about it was, of course, if you look at the cell, and I've shown you a little piece of toy art that my kids and I have put together using these little beads, the cell has to be constructed out of these random parts. But despite that randomness, there is order to the structure of the cell. And so I started to work on this problem of organelle biogenesis when I was a postdoc in Aaron O'Shea's lab at Harvard, where we started to really take seriously this idea of variability in organelle construction, and in terms of really basic properties, like how many organelles are there of a given type in a cell and how big they are. So that's really where I'm coming from here. And to get started, I like to start by thinking about this quote from Jacques Monod, which is, "The dream of every cell is to become two cells." And just to illustrate this, I have this little piece of art. This is a budding yeast cell that's sculpted by a colleague of mine at the University of Chicago, Alan Drummond, who many of you may know. So he has a day job as a scientist, but he does this also on the side. And of course, this dream of a cell to become two cells, if you look at it at the surface of the cell, say the surface of a budding yeast cell, it looks pretty easy to do, right? You have this ball on one side, and it just sort of pooches off another ball on another side. And, it doesn't look like it ought to be all that complicated, but of course we know better. We know that this dream that the cell has of becoming two cells is quite complicated because, for example, in the case of the eukaryotic cell, like we're looking at, the internal structure of the cell is very complicated, right? And so this goal of one cell becoming two isn't just about replicating this little balloon-looking thing and pinching it off into a second balloon. You have to replicate all these internal structures too, or rebuild them from scratch in the daughter cell. So this dream at the surface is actually quite complicated by this internal complexity. And in fact, it gets worse, right? Because the processes by which these mesoscopic structures like organelles are created ultimately depend on the output of giant intricate non-sets of reactions, biochemical reactions. This, for example, I'm sure many of you have seen. It's a chart of metabolism. And the idea is that the cell is able to take in simple inputs like glucose, run this input through this whole chain of complicated reactions, and out of this mess is supposed to come this sort of order that see at that mesoscopic scale. This just illustrate that glucose is pretty simple, but the cells seem pretty complicated. So okay, that dream it's complicated by the structure. It gets worse because the structure emerges itself from a very complicated set of interactions. And then on top of that, it gets much worse, which is that if you look at the details of, let's say, one of these reactions in that giant metabolic scheme, you know that the way that you get from one chemical species to another, these edges in this graph, are ultimately the result of things like enzymes, right? And enzymes are proteins that are encoded by genes. And of course, the process of gene expression itself is very complicated. In fact, not only is it complicated, it's stochastic, right? So this is a image from Dan Larson's group, where they use single molecule fish to label individual messenger RNA molecules that encode for the hexokinase 2 and Gas2 proteins, right? And what you see is two cells here where the number of mRNA molecules for these two different key proteins in the process of metabolism, you see that they're very variable from one cell to another, right? So, but despite this variability at this microscopic scale, we still, do see that cells are able to replicate faithfully. So we're left with this sort of conundrum, which is it seems that under the hood of the cell, it's actually built with very unreliable parts, subject to all kinds of random forces that change its that change the number of molecules that are ultimately going to build these structures. And so the question is, if the cell is built with such unreliable parts, where does this order come from? How does biophysical order emerge from this noisy, nonlinear, interconnected network of reactions? So that's I would say, one of the key motivating questions that drives our own research. Before I go on and introduce you to some of the hypotheses about, or some of the approaches we're taking to understand this very broad question, we're obviously not the only ones studying it. I'll take a second to stop and say, if you have any questions at all, please feel free to interrupt me. This is I really intend this to be very informal, and this is more to I want to learn as much from you as you learn from me. So, so what are some ways to coordinate this process underlying cellular self-assembly that allows you to take these unreliable underlying parts and turn them into this emergent order? So one approach we've taken is to actually measure variability and fluctuations at the scale of organelle biogenesis, and try and understand from just a slightly more abstract view, are there potential mechanisms or processes that we can hypothesize about based on the data that we collect that at least begins to allow us to understand where this order comes from, right? And so I'm going to focus on this first point for most of the talk, which is, can we find some ways that cells can cleverly regulate these processes of self-assembly to at least even out these fluctuations that are sort of brought into the process of organelle biogenesis through these molecular scale fluctuations, and can the cells sort of at least even out this noise over a wide range in, for example, organelle sizes. If I have a little time at the end, I'll get to some more recent work that's very preliminary and we're actively pursuing right now, which is about how you coordinate the self-assembly of these structures with the overall rate of growth of the cell. That's definitely another key way that cells can sort of faithfully replicate themselves. Okay, so just to give you a little idea of the systems we look at to study this very broad question, this is just a cartoon of the eukaryotic cell. And the idea here is we know for a fact that, that some of the organelles in the cell are under all kinds of feedback control, right? It's not that every organelle in the cell is completely going to, that we'd expect them to completely exhibit random variability. For example, the nucleus, for the most part, is under a pretty strong control. Usually cells, unless there's special cells that are syncytial or something, or multinucleated by design, usually cells have one nucleus, right? Similarly, in the process of cell division, we know that, that there are almost always just two centrioles that govern the construction of the mitotic spindle. If you have more than two, you actually get all kinds of strange mitotic defects that lead to things like aneuploidy and cancer. So, so we know that there are examples where you have some sort of feedback control or something to limit these fluctuations. But for organelles that whose biogenesis is at least in part demand driven, so these are organelles that are responsive to, for example, environmental change. We have this very to us simple question, which is, about what kind of variability those organelles would face. I see there's a question about whether organelle assembly noise is something intrinsic to chemistry, or whether there's a way to optimize the biochemistry to eliminate that noise. So I hope the examples I just gave about the nucleus and central mean that there are ways that actually you can eliminate the noise. Those are, I would say, still actively being worked on, exactly what the detailed mechanisms are that lead to, for example, a cell being able to count to 2, to get 2 centrioles and not more or less than 2. The question for these more responsive organelles, the ones that aren't following the sort of something more like a preset program, like mitosis, those are ones that we're really interested in. Okay, so now, of course, in the lab, we don't just draw cartoons, right? We can actually do things like measure, right? We can measure the properties of these organelles. Shown here is a micrograph of a field of yeast cells whose lipid droplets have been fluorescently labeled by fusing the yellow fluorescent protein to a gene called ERG6, which is a major constituent of lipid droplets. And we can actually zoom in on individual cells, right? So we got this cell right there. And we can take a look at some properties of these organelles in great detail, right? So for example, we can do things like count. We can count how many lipid droplets are in this single cell. I can sort of look for bright structures. We do a lot of controls to make sure that our counting is accurate and reasonable. And moreover, we can also measure the sizes of these lipid droplets, right? We can actually just go ahead and count up how many pixels belong to this object versus how many pixels belong to this object. So, so what this allows us to do then is actually capture the kind of statistics that we're after in order to answer questions about how, under what kind of control are these more responsive organelles. And so now we're in a good position to be able to understand some basic principles that we think control these quantitative aspects of organelle biogenesis. Things like how many are there and how big they are, and what are some of the rules that dictate how many there are and how big they are, right? So one thing we did very early on in my own group when we started this project is we were very interested in sort of establishing what kind of constraints we think are operating on organelle biogenesis, right? So, for example, if you wanted a cell to make more organelles, let's say you had a certain number, and let's say some environmental change happened, and you wanted to proliferate the number of organelles in this new environment. Is it the case that the cell has enough resources to just build new organelles sort of willy-nilly with, and those new organelles will have all the same properties as the old ones in terms of their, for example, their sizes, right? Or are cells sort of fundamentally limited in some way in allocating biomass toward this construction project, right? So are cells subject to, for example, a limited pool of building blocks with which to even build these organelles? So that's the first thing that we were interested in, because that, even at the level of averages, that tells us something about what kind of variability the cells can tolerate and what are the impacts of that variability on organelle biogenesis. So the first thing we did was we measured in lots of cells the number of a given organelle. So we focused on 4 organelles in particular: the Golgi, peroxisomes, lipid droplets, and mitochondria. And we measured the average sizes in those cells. And we noticed in each case is that when you look at the average size of the organelle, whether you're looking at Golgi, mitochondria, lipid droplets, or peroxisomes, and you plot that average size as a function of the number of organelles in that subpopulation of cells, you see this strong trade-off between the average size and the number. And this, to us, plus a lot of sort of other modeling and sort of control experiments, led us to conclude that there's a trade-off in some ways between number and size, meaning that maybe there's some pool that's limited, that right? So the more organelles you want, the smaller they're going to be, because you just have a lot of mouths to feed, basically, right? So you're going to run out of material to make a really big organelles if you want a lot of them. So this is the first sort of, I would say, constraint that we saw from our experiments. This is us doing quantitative microscopy on populations of yeast cells. Now, we could have left it right there and said, okay, well, that's an important allocation principle in cells, and but then we saw this paper that really fascinated us about the implications of trying to grow multiple structures from a limited pool in terms of the cell's ability to control organelle properties. And that was this paper from Janne Kondev's group, who, among other groups, showed that if you're trying to grow the organelles and in a controlled fashion from a limited pool of building blocks, that actually leads to really dramatic fluctuations. So I should take a step back and say that one of the reasons why the limiting pool model for being a constraint on organelle biogenesis was so appealing was that it sort of gave rise to a natural way to control organelle size, right? The cell basically synthesizes as many building blocks as it needs, uses them all up, and that that limited pool by itself sets the organelle size, right? And if you just have one organelle, like just the nucleus, for example, then actually this principle works pretty well. If you look at the sort of size distribution of hypothetical organelles, if you have just one organelle from this limited pool of building blocks, you actually do get a pretty tight restriction on the size of that organelle. However, once you have even 2 structures, that size control immediately goes away. In fact, for the case of 2 structures, you get quite the opposite. You get actually a uniform distribution of sizes. You completely lose control over sizes. And so this to us was very striking, as a very striking prediction. And so we got really interested in seeing, in real cells, is it the case that as you, have a multiple number of organelles, do you actually lose size control, right? Is this just a fundamental limit to how precise cells can control their organelle sizes with? So we embarked on this question of using our quantitative imaging data to actually ask, how precisely can cells control the assembly process of its organelles? So before I go on, I wanted to sort of, let's think a little bit first about what growth even means, right? Especially random growth, right? So the idea is, right, we have all these random forces acting underneath that regulate the process of organelle growth. And we have this idea on one side that limiting pool, if you have a limited pool of these building blocks to build those organelles with, it'll lead to all kinds of crazy fluctuations in organelle size at the end of the day. So let's think about sort of some places where trade-offs between organelle size and control of organelle size can arise from. And the analogy I like best is to think about this little boy who I found on the internet blowing a balloon. So let's imagine that you have this boy blowing up the balloon, okay? And imagine that you want this balloon, that the boy wants to blow the balloon to different sizes, right? Just so that it tunes the size of this balloon towards some demand that that he's facing. Okay. So let's imagine there's 3 different sizes that the boy can blow the balloon to: this sort of small size, this medium size, and this really large size. And the question is, if you want to do this as a cell, which is subject to all the usual dictates of statistical mechanics and thermodynamics, how does this actually, how does the boy actually do this? In the way we usually think about. So usually the way we think about these kinds of processes happening is through something like a Poisson process, right? It's a random process where you basically you'll have the boy sort of blow into the balloon in constant amounts, right? Which represents basically constant numbers of molecules, say going towards organelle growth. And what you do is you change the rate at which you blow those, at which you blow the balloon, or the rate at which you add those molecules to the growing organelle in order to get them to be bigger, right? So for example, for this small balloon, you can imagine that the boy gives one little puff. That's what these red marks will indicate. And he'll puff and puff and puff again a little bit later and puff. And at each puff, the balloon size or the organelle size will grow a little bit. Okay. And so you'll, and then in between these puffs, maybe the balloon has a little hole or something, and you have a little bit of air loss, right? And the balance between the puffs of air that the boy is blowing and the slight leakage from the balloon leads to a steady-state size, okay? And the idea is, when we normally think of the physics of random growth, when the boy wants to blow the balloon to a little bit bigger size, what you do is you change the rate at which you puff, right? So now you puff a little more often. The size of the increments is the same, but because you're puffing more often, you get to a higher steady-state size. And similarly, for the biggest one, you have the most puffs, and so you'll get to the biggest size. But the size increments are the same. That's the key property with this kind of Poisson style growth that we think about usually. Okay, so what that prediction, so let's think about what that predicts then, right? What that predicts is that we'll get something very similar to what the Yanev result was about, which is that as the size gets bigger, right? And remember, size getting bigger in our limited pool picture means that the number is getting smaller, right? The number of organelles. As the size gets bigger, the relative fluctuations around that steady state size get smaller, right? Because you have the same magnitude of relative fluctuations, but they're around a much higher average size. So here the relative fluctuations are quite small. Here you have the relative fluctuations being the same size, but they're around a size that's much smaller, right? So now these relative fluctuations are much bigger. So you expect this trade-off basically, where at small average sizes, meaning high numbers of organelles, you will get relatively large fluctuations. Whereas at large sizes, meaning small numbers, you'll get relatively small fluctuations. It should be this fundamental trade-off that you can't break. Okay, and so what I'm going to show you is in the insets of these plots is what we'd expect to see from this Poisson sort of growth model. Okay, so so what I'm plotting here is a property called the coefficient of variation. So it's the standard deviation of the organelle sizes divided by the mean organelle size. So that's it's related to measuring how big those fluctuations are compared to the average size. And I'm going to plot that a function of the size, right? And so what you see in these inset plots are what we expected to see, right? You expect to see that small Golgi apparatus, you get large fluctuations. Whereas at large Golgi, you get small fluctuations. And similarly for the lipid droplets, peroxisomes, mitochondria. But of course, we actually have experimental data that we can actually measure these fluctuations from as a function of the sizes. And to our great surprise, the fluctuations looked nothing like what we expected, except in the case of the mitochondria. So when you're looking at the Golgi, the lipid droplets, the peroxisomes, it appeared as though actually somehow the cells have figured out a way to control the noise in their organelle assembly processes, independent of what size they're being set at. So I'm going to stop here just for a second and see if there are any questions, because this is an important point to think about, because it's a very to us at least, it was a very non-trivial result. How do you decouple the scale of the fluctuations from the size of the organelle itself? Okay, so we stepped back and decided, okay, well, we need to rethink this whole idea of how we think growth is even happening for these organelles. So let's go back to the boy with the, okay. So the boy still wants to be blowing the balloon to different sizes, right? The cell wants to grow its organelles to different sizes. And now we want to understand, okay, where does that actually come from? Where's the what's another way we could try and understand how this growth happens, but in a way where the noise is, insensitive to the size. So of course, when we talked about this the first time, we said that all that changes is the rate at which the puffs happen, but the puff sizes stay the same, right? We said that was the basics of Poisson type growth. Okay, of course, that was an assumption we made. If you actually go and think about how organelle size is controlled, we actually know that that's not exactly necessarily how organelle growth happens. For example, in the Golgi apparatus, right, we know that the Golgi grows through a process of vesicular fusion. And the vesicles that feed the Golgi, they're not always the same size. There's actually, people have measured variable sizes in the Golgi, in these COP2 vesicles, for example, that come from the endoplasmic reticulum to the Golgi. So we were inspired by ideas like that. Like, okay, so what if we relax this? What if we relax this requirement that the sizes of the increments have to be the same, but instead allowed the boy to puff at maybe the same rate, but instead puff different amounts of air into the balloon? It sounds like a really small change, actually, but it actually leads to a really big difference, which is, okay, now if we make a boy puff air into the balloon at the same rate, but change the size of the puffs, you actually get something that looks really different in terms of the fluctuation magnitudes. Now, in order to maintain a certain steady state, the boy has to be blowing more air per puff, right, when he has only the same number of puffs to work with in order to get to a bigger size. So the fluctuation magnitudes actually grow. For the biggest balloons, they grow quite a lot, right? Because now you need to puff quite a lot of air into each individual puff in order to even achieve this bigger size. So now it looks like we have this way where we can relate the fluctuation magnitude to the size of the organelle. And when we do that, we can actually formalize a mathematical model of organelle growth that's quite different from the one that all of us thought about before, as most of us thought about before. So in this new way of looking at organelle growth, what we think is happening is the following. It's a lot like this boy blowing the balloon. So organelle growth proceeds according to two processes. One is characterized by this parameter called the burst frequency, and one is so this is basically the rate at which material is allowed to be transferred to the organelle to have it grow, right? So that's those red marks that I talked about before. The other process that grows the organelles is characterized by a burst size. So that's the size of the growth increment, right? That happens at a rate of alpha. And importantly, what we assume is that this burst size is itself variable. It's maybe the downstream process of those stochastic molecular scale events that were happening that we talked about at the beginning of the talk. Okay, so if we say that there's a burst frequency and there's and the different growth steps happen at a different burst size, then we actually have a way to do two things. One is we can still regulate the size of the organelle, right? We can regulate either by increasing the burst frequency or by increasing the burst size. Both mechanisms will lead to larger organelles. The mean organelle size, in fact, is the product of the burst frequency and the burst size. It's alpha times beta. That's going to be important for later. What's really remarkable, though, is the effect that the burst size and the burst frequency have on the fluctuations, on the variability in organelle size. The two have totally different effects on the variability in organelle size. If you change the burst frequency, that's like the first balloon example we talked about. That's like Poisson growth, where you increase the frequency in order to increase the size of the organelle. And there we see this trade-off between the fluctuations, which we are plotting on the y-axis in this model, and the size, where at small sizes you get large fluctuations, and at large sizes you get small fluctuations. If you just change the burst size, but hold the burst frequency constant, it turns that you can, in fact, get this pattern that we saw in our data, which is that no matter what size you're at, the relative fluctuations actually remain the same. The sort of mathematical underpinnings of this was actually developed in a very different context. It was developed in the context of stochastic gene expression. And this is from a paper, I think in 2006, from Sunny Shi's group, where they showed that a model like this, where you have a burst frequency and a burst size, will lead to a distribution of size that what's called a distribution. Okay. And this gamma distribution has a very particular property, which we already plotted here, which is that this coefficient of variation, this measure of variability we're using, the standard deviation divided by the mean, depends only on the burst frequency. Okay. All right. Everyone's still sort of with me? Okay. Why? Okay. So now, we're proposing a very different picture of how organelle growth happens, right? We're saying that instead of sort of like growing little bits at a time at some pace, you actually have these burst-like processes where you will release material at some constant rate, basically, right? But the size of that material is going to change randomly. Okay. So how do we test this? So lots of ways you can test this. One particular way that we found very satisfying was to look at the following. So if if the goal, so remember, we have statistics like these probability distributions of the organelle sizes for, very distinct organelles, right? We have one set of data for Golgi, one set of data for lipid droplets, one set of data for peroxisomes, which are the ones that follow this flat noise profile, right? It's not like the mitochondria. And one thing to step back and think and remember, to appreciate, is that the molecular mechanisms by which these different organelles are constructed by are vastly different from each other, right? There's really little relationship between the pathways that feed Golgi growth, for example, versus lipid droplet growth. So Golgi, I've already mentioned, are grown by these COP vesicles. Lipid droplets are actually the result of, they actually are like little balloons. They're the result of neutral lipid synthesis enzymes synthesizing lipids and feeding them straight into the lipid droplet and making them bigger. Peroxisomes, they import material from the cytoplasm using post-translational mechanisms. So their growth is also really different. And they also get material from the ER, maybe the mitochondria, they're really different from each other. That's really what I want to emphasize here. And if you plot all these distributions on top of each other, you get this very scattery looking thing, right? Where they don't look very much like each other. Our mathematical model, however, makes a very, very strong prediction for what this data should look like if we look at it just right. Okay, so let's go back to them for a second. The mathematical model says that if I do if I do the following transformations, I should get something sort of almost magical happening. So if I rescale, if I rescale the organelle sizes by this burst size, okay, so now the data look like it all has the same burst size. And then they should, and if I rescale the data by this burst frequency, they should all, so if I take away all the information about the burst frequency, then all these different probability distributions, they should look the same because I've taken away all the organelle specific information, and all I'm left with is this gamma distribution, this sort of non-dimensionalized version of a gamma distribution. So, and what I want to emphasize is how we actually do this rescaling, right? It's completely data driven. There's no fitting parameter at all in any of this, right? So the way we get our burst frequency, this alpha parameter, is we actually just look at the coefficient of variation that we measure, right? The CV, like we said, is equal to 1 over the square root of the burst frequency. So if we know the CV, we know the burst frequency, okay? So we can get that purely through experiment. Moreover, we know the average organelle size, right? And the average organelle size, because we can measure that. And the average organelle size is the product of alpha and beta. And we already said we know alpha, right? Because we got it from the CV. And we know the mean. And since we know the mean, and since we know the alpha, we automatically can get the beta, again, purely through data, no fitting or anything. So if we take those data extracted alpha and beta parameters and do that rescaling I just talked about. So we rescale all the sizes by that burst size, that beta parameter, and we rescale the distributions by the burst frequencies that again we get from data. All of this data should collapse onto this very simple non-dimensional curve, e to the minus s divided by s. This is what that curve looks like, okay? So it doesn't look very much like the data on the left, but if you actually take all these data points and run it through this non-dimensionalization procedure, you see that they completely collapse very nicely onto this non-dimensional curve, which we take as a very stringent test of at least the accuracy of this theory in being able to describe the statistics that we see. So this to us was very satisfying because this, these kinds of non-dimensional data collapses are sort of the thing that physicists at least live for. But there was another test that we wanted to do before we were totally sure that, well, to the extent that we are totally sure of anything, that this is a legit way to look at organelle growth. And one is a slight detail that I hid under the rug a little. And that has to do with the size of the pool. You could have asked yourself, okay, well, you told me that the pool size, that there's a limited pool. So where does this pool even enter into this picture of your burst like growth model of Organelles? So the reason why we were able to use that data collapse in the previous set of plots was we measured that actually, even though the pool is limited in terms of how big organelles can get if you have more of them, it's not so limited that you actually exhaust the pool entirely. You just get this trough, but it's not total exhaustion. What that allows, if as long as you don't actually exhaust the pool, you just limit it, it still allows the organelles to kind of grow independent of each other, right? It's not that they're actually competing for resources, because there's always some pool in the bank to allow a given organelle to grow, but there is this sort of, the burst size, for example, will change from one category to another. If, however, you actually exhaust the pool, right, where if you actually plot the organelle volume, total organelle volume, so this is all the organelle volume in a cell of a given type, as a function of the number of organelles, if you see that it plateaus like this, then you really are running out of material entirely, right? There's just no more pool at all. Now you have a situation where the organelles, if you're in this branch of or in this part of the curve, now the organelles really are competing, right? If I get bigger, you have to get smaller because there's just not enough of pool lying around. So in this picture where if I get bigger, you have to get smaller, what that predicts is that means that the noise, the fluctuation should actually grow in this curve, right? If you're in exhausted limit. That means our relatives are apart from each other, right? So the noise is getting bigger. So we wanted to test this idea experimentally. What we did is tried a couple different tricks. So here's one where we looked at the Golgi. So in the Golgi, before we had what is shown in dark red, that's the wild type curve. So as you increase the number of Golgi, you increase the total volume of Golgi in these cells, but you see that it's a pretty straight line. Okay, so there's no indication of you actually running out of material. However, if you delete this key vesicle protein, this key docking protein at the Golgi called ARF1, that's what's shown in this sort of lighter pink, purple. You see that as you increase the number of Golgi, slope starts to tail off and plateau at high numbers, just like you would see, just as it's an indication to us that the pool truly is running out. So what we predict is that whereas in wild type, you had this flat fluctuation profile where the noise was flat, but in this delete, we'd expect the CVs to grow from that. And that's indeed what we see. So in gray is, in this maroon is the wild type again, so it's flat. But when you do this delete, you see this start to grow above the maroon, just like we expected from the model. Similarly, if we do this experiment in lipid droplets, we did a slight experiment. Here, we actually grew cells in a different medium. It's a medium that is more demanding of lipid droplet biogenesis. So if you grow cells in oleic acid, we know that lipid droplets have to grow to accommodate this, environment, the fatty environment. And what you see again is, in maroon is the wild type. In glucose, this should say glucose, not wild type. They're both wild type. And in, again, this sort of pinkish color, you see what happens in oleic acid media. And again, you see that at high numbers, you start to see this plateau, right? It starts to flatten out a lot here and fall off the curve that you'd expect if it just continued marching up. And again, we see that in the oleic acid case, that the fluctuations grow compared to what we expect from wild type. And we do the same thing in peroxisomes. So again, you get this flattening of this curve. So this one actually just stays pretty flat to start with in oleic acid. And again, we see these fluctuations grow. So this to us was again, more evidence that this idea is this idea of having enough pool around in order to allow the cells to fluctuate, allow the organelles to fluctuate independently is an important way that cells regulate the size variability of their organelles. Okay, I see some questions, so I'm going to let's see if I can. So these are all very good questions. I'm going to come to them at the end. Okay. So the take-home message from this part of the talk really is that that the size of these membranous organelles seem to be limited by a pool of building blocks. I don't know why it's doing this automatically. So that's the first takeaway, that it does seem that we have this trade-off between number and size of organelles. But what's I think really cool that we could do with the statistics that we collect is that we can actually compare them to our intuition for how organelle growth should have proceeded. And we saw a really strong disagreement between that intuition and what we actually measured. And instead, we were forced to proffer a totally different picture of how organelle growth proceeds. We think it occurs bursts of synthesis rather than, as this stochastic bursts of synthesis rather than this sort of even pace. This stochastic burst mechanism that we think exists, it mitigates fluctuations in the construction process. That's one important biological consequence of this. But it also points a way of for a mechanism by which you can sort of timeshare organelle biogenesis. So as long as you're not all bursting at the same time, you can sort of, for example, imagine the limited pool material is membrane. All the organelles need membrane, but this burst-like process makes it so that you're not all drawing from membrane at the same time, right? You could actually time share that membrane demand. So, and that capacity constraints seem to the precision with which organelle size fluctuations can happen. So I'm going to take a pause here for 1 second and answer some of these questions. And Kate, how long have we been going for? I somehow have lost my clock.
It's 11:51.
Okay.
I mean,
okay.
Whatever, not 11, whatever.
Yeah, right. No, it's 51 past the hour. Actually, these chat timestamps are very helpful. Okay, so let me say, before I answer these questions, let me just say one word about where we're trying to take next. I don't have time to dwell on, but it's just to give you an idea of what we're thinking about. So, of course, this first set of experiments I showed you all had to do with looking at individual organelle statistics on their own in isolation, right? I looked just at peroxisome size fluctuations or just at Golgi size fluctuations, so on and so forth. We know very well that this is not, in fact, a set of organelles, right? We know that organelles, in fact, need to be grown in concert with the overall pace of cellular growth. I see the question about this that I'm going to get to in a second. A couple questions, actually. Of course, we also know that organelles don't operate in isolation from each other, right? Like they actually coordinate activities with each other in some way in order to support overall cellular growth. There's entire classes of proteins that people discovered that actually mechanistically implement these, like things like organelle contact sites, for example. Okay, so what we're interested in doing is we're trying to, one future direction that we're pushing this work into is trying to understand so we know that there's some variability in organelle size and number of statistics, right? Even for identically grown isogenic cells. So one thing we're interested in is seeing whether that variability is something that the cell sort of couples to each other. So do they coordinate the biogenesis of its various organelles and sort of try to limit the interorganelle variability? Or is it the case that these organelles aren't just fluctuating independently of each other, and this is just presents some sort of fundamental limitation to how precisely cells can control their makeup? I'll skip this. One way we're thinking of doing this is actually develop tools in order to look at all these organelles simultaneously. So we've developed these hyperspectral imaging tools using some simple machine learning idea to actually make cells that look like this, for example. So I promise you in this micrograph, there are 4 distinct organelle types labeled. Very hard to tell because, of course, our palette of fluorescent proteins is relatively poor in terms of the overlap, spectral overlap between different proteins. So we did some work to actually try and resolve all these spectrally overlapping proteins. And if you sort of run these kinds of images through a neural network trained by by Xixing Gang from our lab, you see that you get these very nice spectrally resolved. These are obviously false colored, but you can actually tell all these different organelles from each other in the same cell, right? And we've gotten this to the point where we actually look at 6 or 7 organelles simultaneously in cells. And what this allows us to do is to make plots that look like this. So each of these represents an individual cell. And for each cell, we can do things like extract out the number of lipid droplets or the number of mitochondria or the size of the ER in that cell. And now within this giant data set of single cells, we can look for patterns. We can look for patterns of coordination between these different organelles. So in particular, for example, you can look at covariances between different organelles. We do sort of things like principal components analysis to look for collective modes of organelle biogenesis to see if there are particular sets of organelles that the cells regulate together in order to do things like support increased growth. And so this is a lot of what we're spending our time doing now, is to try and understand collective effects of collective regulation of biogenesis in support of physiological demands like growth. Okay, that was quick, but this is just an idea of sketching out things we're interested in doing right now. So really, I think that one of the big take-home messages from all this is we can actually fruitfully use even simple models inspired by sort of the physics of random growth to at least propose mechanisms for how these processes are actually happening at a sort of more abstract level. Of course, there's tons of work to do, right? We still have to, I haven't said anything about the actual molecular mechanisms that drive, for example, burst frequency versus burst size, things like, I mean, we're sort of all, if you take that idea very seriously, it's almost like there's 2 different processes going on that we have to uncover players for. So that's definitely one major thing that we would still want to do. But nevertheless, it's important to pause and appreciate the fact that that these simple mathematical ideas actually do give us some insight into how things could be working. And I want to stop with acknowledgements for people people in the group who did a lot of this work. So the work I presented here was done by Kian Dokt, Panchana Amiri, and Asa Kalish. But everyone contributes to all of these different projects. So I thank them greatly for all their hard work and ideas. Okay, and that's really the end of the talk. And I'll start trying to answer some questions. Feel free to keep these questions along. I'll start with some of the, Emma asked, why do you use eukaryotic cells instead of simpler bacteria? So I think when I started this work, the sort of number of membrane-bound organelles, and even today probably, there aren't a ton of examples of membrane-bound organelles in bacteria. There are some that maybe people, there are definitely protein compartments. Now it looks like there's even these membrane-less organelles in bacteria, but that's really why we picked the simplest eukaryotics we could think of as far as being a model organelle with lots of knowledge of it, in budding yeast. So we went as close as we could to bacteria, but still preserve the complexity of all the different organelles being present. Instead of organelles, could you do this research on whole bacteria, study bacteria? Absolutely, you can definitely do this. There's a whole field of research that studies bacterial size in very much similar way. I could give you probably names off my head, but I'd leave out 20 more who are doing equally interesting work. So, I'll give you some sort of keywords. You can go look up things about adders and sizers. All of those ideas that you've maybe heard of, all could definitely apply to this. For mitochondria, could you work with yeast mitochondria? You'd have more flexibility since they're not crucial. Yes, so we actually do. I agree that they're not crucial for, we could actually work with petite phenotype yeast. That's the name for when you delete the, so the mitochondrial genome apparently is important. The mitochondria themselves, no, sorry, not mitochondria, the mitochondria structure is important. I think the hypothesis, the lead hypothesis still for the construction, for the synthesis of iron sulfur cluster proteins. So you do need them around, but you can get rid of their basically their oxidative capacity, I think, and they still survive. So that could be an interesting, especially for this multi-organelle, collective organelle stuff, those could be really interesting mutants to look at. Is organelle size tied to cell size, and will a larger cell have bigger or just more organelles? There's again a large literature actually about the coupling between cell size and organelle size. For many of these organelles, there's a well-demonstrated scaling relationship between cell size and organelle size. On average, I should say. I should definitely put that proviso in, on average. When you actually look at these fluctuations, you can in fact recover these average relationships for cell size and to some extent number, but the fluctuations are around those relationships are pretty large in our hands at least. So so there is a relationship, but I think that the variability is also pretty substantial. Is organelle growth limited by cell growth age and division? Oh, sorry. Yes. Do older cells have longer to wait? This is a question from Bernie. Do older cells have longer to wait for their burst and growth? This is a great question. We're really interested in exploring it with our sort of new ideas on how organelle growth proceeds. There definitely is a relationship between organelle growth, at least, yeah, and cell age and division. So there's a great story about how mitochondrial defects that are downstream of a vacuolar defect, which is downstream of a plasma membrane defect, which is a result of the aging process. This is from Dan Gotchling's group. That I think is if you haven't read those papers, you really should do yourself a favor and do that now, because that's a great example of how organelle biogenesis is limited by impacted by age. Okay, so from Shritama, Bal, great talk. Does this mean one way of growing organelle could be like tuning mechanical properties of organelle membranes? That's a very interesting idea. We have not thought about that essentially at all. I could see that, right? So if you limit the rate of uptake of whatever material is being transferred by, for example, changing the membrane properties, I could see that being plausible. I don't have any good evidence for it, but it definitely sounds plausible. Lastly, why do these organ studies have to be membrane bound? So they don't. We actually have looked at statistics, some of the membraneless organelles. I don't remember what the status of those experiments are. If I'm not mistaken, we saw similar statistics with those, but we don't have a good explanation for why that would be in terms of these flat noise profiles. But I'd have to go back and actually check our old data, but you're correct. They don't have to membrane bound. The reason we were particularly interested in membrane bound ones is because of these collective effects that we know of a lot of examples of different organelles working together. And so all that early stuff I was talking about was with an eye toward these multi-organelle systems that we're working on now. But yeah, these statistics that we talked about could be done with p-bodies and stress granules and things like that. Any other questions? I hope those were reasonable answers.
Thank you very much. I think I don't see any other questions. I have a question myself. Do you think we'll be able to, if we have an artificial organelle, something like a vesicle inside a vesicle, assuming the cell-free protein expression is also stochastic, will they also grow in bursts, or because it's in vitro, we'll be able to do it less with less variability, with a little more control?
That's so. I think really what the question there is yeah, what is the mechanism that leads to these burst-like profiles that we think are happening, right? Like,
yeah.
I mean, so it depends. So it depends on how you grow the organelle. I think that's maybe my would be my take. So if the organelle growth is through uptake of a protein, say from the cytoplasm of this synthetic cell, then if you do a good job of controlling the synthesis of those proteins, let's say, yeah, I could see this basically being, you know, the fluctuations will just follow the Poisson limit, basically, right? And so if the if your expression of protein is very weak, of course, then you will get, like, you know, larger fluctuations. But if you make it high enough, then those will substantially diminish. If the growth is occurring through something more like the Golgi, and maybe peroxisomes, that's a controversial case. We don't know much about how peroxisomes grow, turns out. Then it comes down to how well can you control the distribution of sizes of these vesicles. That becomes a little bit of a harder problem, probably because that itself is a multi-step process, right? So I think that's what it boils down to, probably, is what's actually causing the organelle to grow. And so for a synthetic case, because you have control over that step, I think you can beat these limits that we're talking about.
Yeah, that's interesting. I guess we have to make the artificial organelle first.
Yeah, right.
Minor
but
issue.
That's the sort of thing that I'd love to hear about all these things, because that's part of why I was very happy to come and join you all, because I think those kinds of investigations could be really important in figuring out just the basic physical mechanisms going on here.
Yeah, and help us then rebuild those mechanisms in vitro.
Right, exactly.
All right, if there are no other questions, thank you very much, Shankar, and I'll post the recording online.
Okay, thank you so much. And if any of you are ever in St. Louis, just come say hi.
Thanks a lot, and thanks everyone. Bye.
Bye.